Part of Task Force 10 had to do with whole food testing. We already mentioned this morning that we have two types of changes, the intentional ones, and the prime concerns there are toxicity that Joe talked about, allergenicity that Ron talked about, and then we get to those unintentional ones, the ones that we primarily do by compositional analysis and agronomic equivalency. But there's still some jurisdictions who say, well, what if maybe your compositional analysis did not pick it up? What if it still did not affect the equivalency? So what if there is something still there that all the other tests have not caught? So the idea is then to do whole food toxicity studies, where they take the techniques that were done for isolated defined chemicals and apply it to a whole food. And that means that instead of feeding the rat the isolated protein that you were mentioning in this case, you would feed them the whole soybean, the whole corn, or whatever is in it. So to ask the question, is this really a good way to go after anything that we might have missed? We started by looking at the past. The idea of whole food toxicity studies comes out of the use of irradiation, radiated foods, where foods are irradiated in order to kill any pathogens that have that may have been present. So it's a way to increase safety. But at the same time, people said, well, maybe you're irradiating food and there's this unknown things that you're creating in there and they might be dangerous. So how are you going to assess them? So the concern was large enough that there's a lot of literature on chemistry and toxicology of irradiated foods. There's a lot of information on the risk assessment that has been done. And there's also a lot of people that criticize the studies to say that they really didn't show much. So we started by looking at that and then applied all that to whole food studies using genetically modified crops. And again, what was done, how the study was, the limitations. And then something that was probably not a big deal when people were doing irradiation studies is animal ethics has become very important. So this is the task force that Joe was mentioning, that looking at protein safety. And the results have been, the manuscript has been accepted for publication, and as Joe said, should be out in a month or two. So, going back in history, and really history is repeating itself in that the same arguments are here. Well, maybe it's not as nutritious, or what are the unintended, unknown toxic products? I think the only different, well, actually, in the past, people were afraid that if you irradiate a food, it would become radioactive, and now people are afraid that if you genetically modify a food, you can become genetically modified by eating it. So the concerns are very similar. And those concerns did really delay the adoption of irradiated food, and it's still very rare in many places. So again, looking in the past, chemistry and toxicology studies of the foods, and those studies predicted then that one could use a purely analytical, laboratory analytical process to determine if they were wholesome or if there might be any health risks from them. And in general, there are changes from irradiation, but there's also changes from cooking the food, and people don't worry that much about those. And the changes we were making by irradiating foods, or I should say they were making by irradiating foods, were fewer than those made by cooking. And when they used, again, analytical methods in the lab to look at what was being produced, they were producing things that were naturally present or also found in foods that were processed by other means. They really didn't find anything that was unique or novel. Nevertheless, many subchronic and chronic animal feeding studies were conducted about over a 50-year period. They looked for teratogenetic effects, mutagenic effects, carcinogenic endpoints, and quite a few thousand animals have given up their life in the name of the cause. And these range from rats and mice to dogs and primates and chickens and quail and whatnot. And in all those studies, they never found anything that was unpredictable based on the chemicals being formed. Nothing, if there was an inherent toxic, fine, but there was no new toxins that were being created. And whenever they found adverse effects, they were because either the nutrients had degraded or there were some other nutritional deficiencies. They weren't giving them a balanced diet or what have you. So again, a lot of parallels to the GM situation. When the whole strategy was reviewed by this joint group from FAO and the International Atomic Energy Agency and World Health Organization, they concluded that you can use the chemical data present allowed you to extrapolate and determine if food was wholesome or not. And even though radiation caused chemical bonds to break and other new ones to form, if you understand what the compounds are forming and unforming, that you can understand whether the food is wholesome or not, because then you can compare them to what else is out there and you can identify any that you might want to take a closer look at. However, what this one review really did not talk about was the role that animals played in the safety studies. It talks a lot about what the chemical analytical studies did. But when it comes to animals, it says none of the toxicological studies with animals had produced evidence of adverse effects. Now, you can look at this in two ways. One way to look at it is saying, oh, irradiation is safe and therefore there were no bad things for an animal to detect. Or you can say, well, maybe had there been something bad, the animal can't detect it. And this is a study from about 30 years ago that was just saying that if there was something really harmful in the food, you're not going to detect it by feeding whole foods to animals. Number one, you have no clue what you're looking for. You have no clue what type of chemical it is, if it's there, how much it's there. You're really operating blindly. Secondly, as has been said, I think by every speaker, dose makes the poison. The classical toxicology is you escalate dose, and as you escalate dose, you expect to see a dose response. With whole foods, once an animal is full, you cannot keep on stuffing more food in it. So you really cannot do the classic dose response with an animal. And furthermore, if we're looking for the other thing is that if you give an animal too much of one food, they don't have a balanced diet, which in turn creates additional problems. You'll get symptoms from malnutrition that have nothing to do with the safety of the food. And finally, the type of effects we're looking for are usually small, and really it's not possible to make a sufficiently large experiment that would give us any degree of confidence that those small effects were due to the presence of a radiolytic product or just random background noise or something else. So the whole food studies, as applied to the GM crops, again, is the last safety screen we have in case we missed anything. And from there, extrapolate that if it's safe for the rat or the mouse, it's going to be safe for humans as well. So whereas one half of the Task Force 10 was working on protein, and that's the work that Joe described, the rest of us were reviewing the literature on whole food studies. And there's about 50 published studies on this, 40 of which are on rodents. And we included all the controversial ones. For example, there's the one that everybody's familiar with out of Scotland, the push tie one that said that rats, the GM was bad with the rats. Well, he didn't control for the glycoalkaloids in the potatoes. So, but we reviewed all the ones that were controversial. So most of the studies were subchronic or acute. I think went up to 90 days. All of them used OECD guideline 408 for rodent studies. The majority of them used rats. And this is just, I'm not going to go over this table, but just to point out that, we looked at how many animals were per group and their gender. We looked at how many doses they had, high dose, low dose. Again, you can't do very many. Reference groups. With toxicology, you want to have other reference groups to see how they behave. You have historical reference groups to see how the results have been in that particular laboratory. Whether the control was an isogenic or a formulated diet and whatnot, and then the percent at which it was added. In general, most of the studies use somewhere between 15 to 70% of the whole food, depending on the crop, and most of them just had 2 dose groups. Most of them used near isogenic crops as a comparator. And I should say, there's some, for example, they were isogenic, but ones were grown in, say, Canada, and the other one was grown in Austria type of deals, which I think invalidates them being isogenic. Again, there were a lot of reference comparators, just traditional laboratory diets, other crop varieties and whatnot. And I mentioned the historical results for health endpoints. And interpretation was really based on statistical comparisons, multiple statistical comparisons, and at the end, a weight of the evidence, as what sort of was described for the allergenicity. There are also several published studies using domestic animals, mostly with bovines, but there's some with swine, poultry, with some, I think we saw a couple with fish. And these other animal studies, you really cannot call them a toxicological study. They just don't meet all the criteria. What they do serve, though, is as you can say that your GM is not nutritionally inferior to the conventional crop, but to call them a toxicological study is really a stretch. Here's just an example of the ones done on cattle, how long the studies were for, from 3 weeks all the way to 84 days, controlled diets, reference groups, and so forth. Reviewing all of what the published literature, none of the properly conducted whole food studies conducted on the commercialized crops identified any adverse effect. And properly conducted, as I said, if you grow one isoline in Canada and one in Europe, because of the variation you get, that invalidates that. If you do not measure key compounds like glycoalkaloids or isoflavones that are biologically active, that can also invalidate results. But there was really nothing then that was, because all the results were negative, there's nothing that would question the adequacy of the comparative risk assessment leading to substantial equivalence that we've talked about this morning in the agronomic and compositional analyses. So essentially we can say that there's pretty much complete agreement between an animal study and the comparative safety assessment. But this gets us back, though, to the same issue we had with the radiolytic studies. Does that mean that whole food toxicity studies are predictive of safety? All the crops that were fed were safe? No, it does not. What it really reflects is that, number one, as I mentioned this morning, the potential for an accidental generation of an unknown toxic substance in a crop, GM or otherwise, is very low. We've never seen it happen. That's how low it is. And secondly, that the toxicity study is not very sensitive. It cannot, feeding animals is not going to find an unknown compound that's very high, that's highly toxic at low levels. It will find a highly toxic compound at high levels, but in that case, we can find it analytically as well. So it's not going to find unknowns of any, you know, if the level of toxicity is low in your unknown compound, you're not going to find it regardless of dose that you can feed it. The bottom line is a rat is simply not a laboratory instrument. So why are they so insensitive? The reasons that already have been mentioned. The normal sources that give toxicological studies their power cannot be done. You cannot escalate the dose. You cannot determine what the maximum tolerated dose is. So, you know, all the doses we're giving are in the no effect level, and you have to interpret that if you don't know what the effect level is. You don't know what chemical you're testing, so you don't know anything about the properties of that chemical. If toxicological studies use dose escalation and pharmacokinetics to make up for other deficiencies, small group sizes or species-related insensitivity, you know, what's toxic to one may not be toxic to another. Tomatoes can be quite toxic to rats. So you can't really do a rat study with tomatoes, even though they're fine for us. Or just a genetic, individual responses probably based on genetics of whatever you're testing. And then I get back to those that I mentioned cause harm. The studies say. There's some that are very well conducted studies, but their interpretation was questionable. One of the things is, remember, we keep going back to, we're looking for 5% differences. Some of the, that means 5% are going to be false positives. Some of the original Seralini papers really got on those false positives, those 5%. So, and there were, even when the differences were well within normal ranges, as long as the difference is within the normal range, it gets back to statistical versus biological relevance. Then there are other studies. I think all of you saw this picture out of Russia. That one poor rat was fed GM and the other one was not. And you can tell, I'm sure you can tell which is the poor victim here. These just simply do not meet any standards. It turned out in this particular study, the large rat was being fed formulated lab chow by Purina that was, I think, about 80% protein, whereas the GM-fed rat was being fed a home-formulated mix that turned out to be about, I think it was, had half the protein level. So there's, so that type of grave mistake is there. Or they simply don't abide by the OECD guidelines. And some of the other flaws I mentioned about the source of the materials not really being isogenic or grown under very different conditions, what have you. So these studies that show adverse effects have not used proper diets or comparators. Isoflavones in soybean, I showed you this morning how variable they can be. Well, so like in particular study, the one that showed that the liver cell problems or the unexplained changes in testicular cells could very well be due to phytoestrogens, which were not measured at all. So is it ethical to be asking animals to sacrifice their life to science if the biology doesn't give us a biologically realistic method by which a toxic substance is going to be produced? And if you've done all the laboratory and field studies and those raised no concerns, and if we go into this knowing that they are not sensitive enough to detect potentially toxic unknown substances in food anyway, And furthermore, if we know that it really does not increase the safety of the product, it does not make consumers any safer, it might make them feel safer with toxicity testing, then there might be a role. And even then, though, we're not saying just do a whole food study, but maybe isolate a food component that's been enriched for whatever novel substance there may be. So to conclude, whole food studies simply are not sufficiently sensitive to detect unknown new toxins in a crop plant. And increasing the duration of a whole food study from 28 or 90 days or to even longer period does not correct for the inherent limitations. And EFSA itself has said as much. So if 90 days is not going to do it for you, you cannot fix it by going to a 2-year study, which is what the European Commission directed EFSA to do. Stick to the lab. The current compositional analysis is robust enough, combined with the toxicology and the allergenicity for whole food safety studies. But toxicology studies, perhaps with a fraction of the food, not the whole food, may be useful on novel substances or metabolites that were introduced intentionally or unintentionally into a crop. So those were the recommendations. And incidentally, right after, the lessons of irradiated foods should be heated. About 2 weeks ago, this publication came out, and it reached the exact same conclusion that we did. So that one is available. And just a parting thought. It is much more convincing to be able to state that certain likely effects have been searched for and found absent than to delude yourself that you didn't really know what you were looking for, but you didn't find it, so it doesn't matter. So with that, this talk was actually put together by Genevieve Bondi, and she gave it at the OECD meeting in Paris earlier this year. So I needed to acknowledge that and then say thanks, and I think there's still time for questions.